Hello friends, how are you? My name is Rizwan Hafeez and this is lecture number 3 on cancer. In the previous two videos, we have talked about the hallmark of cancer and their genetic aspects as well. In the lecture number 2, I talked about proto-oncogene along with different examples. In today's video, I will be talking about the tumor suppressor genes. Well, while explaining about the before explaining about the tumor suppressor gene, first of all, I would like to give you a brief summary on the proto-oncogene, and then I will move toward the tumor suppressor gene. Proto-oncogenes in the previous video I talked about are those genes which are responsible for the cell proliferation, cell division, cell replication, which are responsible for taking cell from G1 phase into S phase. Those genes are basically called as proto-oncogenes. I will explain. In this cycle, you can see in G1 phase, the enzymes required for the DNA replication, you know that DNA replication takes place in the S phase. So all the enzymes required for the DNA replication will be synthesized in the G1 phase, obviously through the process of transcription and translation. It means the genes of those enzymes will express, will transcribe, will translate into the G1 phase, and when the enzyme will be synthesized completely, the S phase, DNA replication phase will start. Okay, assume that these are the set of genes who are basically responsible for the DNA replication, and their transcription will take place in the G1 phase. Now, what are proto-oncogenes? Those set of genes who will stimulate these genes and who will stimulate the process of transcription and who will help the cell to enter into S phase from the G1 phase will be called as proto-oncogenes. It means they are helping the cells to enter into S phase from the G1 phase. So it means they are basically helping the cell for the proliferation. They are stimulating proliferative enzymes in the cell. So it means all the set of genes who are responsible for cell proliferation, who will increase the cell proliferation, cell division and DNA replication, those set of genes will be called as proto-oncogenes. For example, growth hormone receptor, RAS protein, MYC protein, all the function of all these genes has been explained in my previous video. Okay. Now, what are tumor suppressor genes? Obviously, if a cell is proliferating, after some time, it needs to be stopped. Otherwise, if it will keep on proliferating, obviously, you know that this is a pathological, it's not physiological. Okay. So, there must be a kind of molecular break who are responsible to stop the proliferation, to stop the activation and expression of these genes. And those genes who are molecular breaks or who are basically inhibiting these genes are called as tumor suppressor genes. For example, these are the set of genes who are inhibiting, who are giving a negative signal to these set of genes are basically called as tumor suppressor genes. Obviously, they are not basically suppressing the tumor. In fact, they are just inhibiting the cell proliferation and cell division. But in fact, later on it was discovered that these genes are basically responsible to inhibit the tumor as well. So they was named as a tumor suppressor genes. Clear? Okay. Now you can see few examples of tumor suppressor genes, which include RB genes, P53 genes, that is very important. P53 is also called as guardian of genome. I will explain in the later part of the video why it is called as guardian of genome. Similarly, CDKI, GTPase, APC, and there are some other examples as well for the tumor suppressor genes. In the previous video, I also talked about that if there is gain of function mutation in the proliferation proto-oncogenes. We know that what are proto-oncogenes who will increase the proliferation. So if the function of these genes has increased, so it means they will increase more proliferation. So obviously, if the proliferation will increase a lot, it may lead to the cancer. Getting it? Similarly, as these genes were basically suppressing the proliferation, they were inhibiting the proliferation. So if they lost their function, now they will not be able to put brake on the running car, and the car will keep on running, and until the accident will happen. And then accident will happen, and that accident is basically cancer. Getting it? So it means if these molecular brake fails, they lost their function. They lost their function. Loss of functional mutation happen in tumor suppressor gene. Again, cancer may happen. Cancer may occur. But if the gain of function mutation occur, obviously they are basically molecular brakes. If their function increase, they will inhibit more powerfully. Now the chances of cancer are very low. So to develop cancer, there should be mutation, but the mutation should be loss of function mutation in the tumor suppressor gene and gain of function mutation in the proto-oncogenes. Gain of function mutation in the proto-oncogenes. Well, in the previous video, some students were asking about the difference between proto-oncogene and oncogene. Well, it's very simple. Proto-oncogene, that will stimulate the proliferation, and the mutated form of proto-oncogenes are called as oncogenes. For example, RAS gene is basically a proto-oncogene. But if there is a mutation, gain of function mutation, now this is a mutated form. Now it can lead toward the cancer. So now this RAS gene that is mutated will be called as oncogenes. Clear? Okay, now let's talk about the tumor suppressor gene. Here are some examples I have explained. Now first of all, I will talk about the RB gene, retinoblastoma. Well, I have explained that these are the set of genes who are responsible for the DNA replication. When they will get transcribed, DNA replication will take place and cell will enter into S phase from the G1 phase. But what will happen? For the transcription of these genes, you need a transcription factor. And there is a family of transcription factor that is responsible for the transcription of these genes. And the name of that family is E2F. E2F family. But there is a protein called as RB protein produced from a RB gene. This protein is not letting E2F to perform its function properly. It is always holding E2F. It is all and not allowing E2F to go and transcribe these genes. Okay, so it means this RB gene is basically inhibiting the proliferation or stimulating the proliferation. Obviously, by binding the transcription factor and not allowing them to transcribe these genes, it is basically inhibiting the proliferation. And the genes that are basically inhibiting the proliferation are called as proto-oncogenes and tumor suppressor gene. Obviously, tumor suppressor gene. So this RB gene is basically a tumor suppressor gene. Clear? Now we know that these cyclin dependent kinase, they are responsible for the phosphorylation. They will add lots of phosphate groups on different amino acid of this protein. They are basically responsible for the phosphorylation of this RB protein. And when this RB protein will get phosphorylated, it will become deactivated. It will become inactive. It will become inactive, and now it will not be able to hold E2F more. And now E2F will be free. It will bind on these genes and start the process of transcription. So it means this CDK is basically stimulating the process of transcription of these genes, and it is basically stimulating the cell to get enter into S phase from the G1 phase. So these CDK genes, these are the genes of CDK. These CDK genes are tumor suppressor or proto-oncogenes. Obviously, they are proto-oncogenes because they are stimulating the proliferation of cells, while RB gene is basically inhibiting the proliferation of cells. Clear? Now, assume that there is a mutation. I have explained, to develop cancer, in the tumor suppressor genes, there should be loss of function mutation. Now, what is the function of these RB genes, these RB protein? to hold the E2F factor and not to allow them to go and transcribe the gene. It will not let E2F to go and transcribe the gene because its function is to hold the E2F. Now, if there is a loss of function mutation, there is loss of function mutation. Now, this RB protein will not, it has lost its function. Now, this RB protein will not be able to hold E2F properly. Now, E2F transcription factor is always free. It will always keep on transcribing these genes and cell cycle will keep on working and the proliferation will increase. Cell proliferation, cell division will increase and this may lead to the cancer. This thing may lead to the cancer. So it means to develop cancer, there should be loss of function mutation in the tumor suppressor genes. Clear? Now, if we talk about the retinoblastoma, mutation in the retinoblastoma, well, you know that for each gene there are basically 2 alleles on each of the chromosomes. Okay. To develop cancer, I am talking about the tumor suppressor gene. Mutation on both of the chromosome is important. If there is mutation, for example, this red line is showing the mutation. If there is mutation in the one chromosome, another chromosome is intact, RB protein will function properly. We will not consider it is as a mutated protein. Okay, so it means mutation on both of the alleles is important to develop cancer in terms of tumor suppressor genes. So now, for example, there are 2 types of retinoblastoma. The one is hereditary and second is sporadic. In hereditary retinoblastoma, what will happen? The mutation on the one chromosome will be present at the time of birth, and after some time, mutation on the second chromosome also happened because of radiation or some carcinogenic substances or some free radicals or whatever. And now when this mutation will occur, now both of the chromosome has been mutated, both of the chromosome has been damaged. Now this thing will lead to the development of cancer. Now RB genes will not be able to produce a normal RB proteins. Okay, and this type of cancer is called as hereditary retinoblastoma. In sporadic, what will happen? Both the alleles are normal. Both the alleles are normal. After some time, one of the gene get mutated, still the patient is normal. Still the individual is normal because both of the alleles should be damaged. After some other time, the second allele also get mutated. Now this mutated gene will produce a mutated protein that will not be able to function properly, and this is called as sporadic retinoblastoma. That is very rare because 2-time mutation is required. Mutation on both of the chromosome is required, but in this case one chromosome is already mutated. Okay, so this is common, but this is very rare. Clear? Well, now let's talk about another very important protein, that is P-53 protein, also called as guardian of genome. It will guard, it will protect the whole genome. How it will protect the whole genome, I will explain right after a minute. Now, for example, a mutation has been taken place in the DNA. Okay, you know that against A, T will be placed, against G, C will be placed. So you know that against A there is a T, against C there is a G, against T there is a A, against G there is a C. But unfortunately, a point mutation has been taken place, and against G, A has been placed over there. And we know that this is a mistake. It's not a normal sequence. It's a wrong sequence. Against G there should be C. Now, if there is any mutation, what will happen? A protein ATM, that is basically responsible to identify the DNA damages, and once it will identify, it will activate some other proteins to repair this damage, like P-53, like breast cancer type 1 genes and some other gene as well, so that repair can be done. Okay, it will activate NER and BER pathway. You know that these are basically DNA repairing pathway. So it will activate all those pathways that are required for the DNA repairing. And so if this ATM detect a mutation, what will happen? It will also activate a P-53. P-53 is most of the time it is sleeping. It will get activated only when there is a mutation. Okay, and who will awake up this genes? ATM. Okay. Now, the first thing which P-53 is going to do, it will arrest the cell cycle. It will suddenly stop the cell division. It will suddenly stop the cell division, so that things can be the damage can be repaired. Damage can be repaired. Now what will happen? This P-53 suddenly it will get transcribed and it will make P-53 protein, and this P-53 protein will activate cyclin dependent kinase inhibitor genes, and when this gene will be activated, it will be expressed, CDKI will be formed. Cyclin dependent kinase inhibitor protein will be formed. Now this cyclin dependent kinase inhibitor, it will go and it will block this cyclin dependent kinase. Once the cyclin dependent kinase will be blocked, the phosphorylation of RB protein will not take place. It means if phosphorylation of RB protein will not take place, RB protein will not allow E2F to go and transcribe these genes. So the process of transcription and the process of moving from G1 to S phase will stop. So first of all, cell cycle will be stopped by activating CDKI. Clear? The second thing, it will go and it will, sorry, not this gene, the product of this gene, that is the protein P-53. It will go and activate DNA repair genes. These are the DNA repair gene. Nucleases, exonucleases, endonucleases. Okay, it will repair, it will activate DNA repair genes. Now DNA repair genes, they will come and they will break this phosphodiester bond. Adenosine will be removed and a true nucleotide, that is C, will be placed over there. Now mutation has been removed. Once the mutation has been removed, now this ATM will not send any further signals to the P-53 genes. So it means now P-53 genes will activate, it will not receive any kind of other signal. What it will do? It will activate, it will stimulate another genes that is called as MDM2. MDM2. It will stimulate another protein, another genes, MDM2, and its product MDM2 will destroy and dissociate this P-53 protein because it is of no use. All the mutation has been repaired. Now we do not need to arrest the cell cycle. Now once the P-53 protein has been destroyed, it will not activate CDKI and CDKI expression will reduce and it will not be formed further. Now what will happen? It will not be able to inhibit the CDK and the CDK will keep on phosphorylating. It will allow E2F to go and transcribe this gene and cell will keep on getting entered into S phase from the G1 phase. So the cell cycle will again start. So what did P-53 do? First of all, it arrested the cell cycle and then it stimulated the DNA repair genes. Repair was done. ATM didn't send any kind of signal to a P-53 for the mutation because the mutation has been removed. Now when P-53 will not receive any kind of signal, it will activate MDM2 genes. MDM2 genes will make a product, its protein MDM2 protein that will dissociate and destroy the P-53 gene, P-53 protein, and now this P-53 protein will not be able to stimulate anymore this CDKI. Okay. Now there is another very important function. For example, the repair is too much The repair is too much that it cannot be repaired by these DNA repairing genes. This DNA cannot be repaired by these DNA repairing gene. Now what will happen? When this ATM is keep on sending the signal that repair is not being done, now what will happen? This P-53 is going to do another very important function. In the previous video we talked about that the cancer cell will not be able to commit suicide. If they commit suicide, it's good for our body. Obviously, our mutated cells should die, but cancer cell will develop a mechanism to enhance the life of their cell. So what will happen now? This P-53, what will it do? It will, when this P-53 will receive a message that the repair mutation is a permanent and it has not been removed by DNA repair genes. Now what will it do? It will go and activate apoptotic genes. Pro-apoptotic genes, like those genes which are responsible for inducing apoptosis in the cell, programmed cell death. So now this P-53 will lead a cell to a programmed cell death, so that these mutations cannot transfer to the daughter cell. Because if these mutations transfer to the daughter cell, and it means this thing will lead to the cancer. So this is the reason. First of all, P-53 will arrest the growth, arrest the cell cycle. Then it will try to repair the genes, repair the damage. If the damage is not repairable, it will activate pro-apoptotic proteins, pro-apoptotic genes, and it will make the cell to commit suicide, so that mutation cannot be transferred to the daughter cells. Clear? So it will activate. This is the reason this P-53 is called as guardian of genome. Guardian of genome. Now assume there is a mutation in P-53 gene. If there is a mutation in P-53 gene, now it means P-53 gene will not be able to work properly. It will not be able to arrest the cell cycle. It will not be able to activate the DNA repair. And the worst thing is, it will not be able to make the cell commit suicide. It will not be able to activate the pathway that lead to the programmed cell death. The mutated cell. The cell has been mutated and its life will increase as well. So it means all the mutation will transfer to the daughter cell, and that's not good for our health. It may lead to the cancer. So this is the reason this is very important genes, and a huge number of cancers has been discovered that happened because of the mutation in P-53 gene. P-53 gene, MYC gene, and this RAS gene, they play a very important role in developing the cancer. Okay. Well, now let's talk about we have talked about RB gene, we have talked about P-53 and CDKI. Obviously, CDKI was basically inhibiting the cell cycle by inhibiting the cyclin dependent kinase. So it means as CDKI was inhibiting the cell proliferation, so it means it is also a tumor suppressor gene, and the mutation in CDKI can also lead to the cancer. Okay. Now there is another very important protein, GTPase. Now let's talk about the RAS. Well, in the previous video we have talked about that when the growth hormone will act on its receptor, this RAS enzyme will get activated. RAS enzyme will get activated, and when GDP will be removed and GTP will be attached. And when the GTP is attached, this RAS protein is considered as active. And when the GTP was attached, this RAS protein was considered as inactive. Okay. Now, when the growth hormone will come, it binds to its receptor. There is a conformational change in the receptor that will lead to the conformational change in the RAS protein, and that will lead, it will remove the GDP and it will attach the GTP. Here you can see 3 phosphate groups. Guanosine triphosphate will be attacked. And when the guanosine triphosphate will be attacked, now this RAS protein is active, and through the two pathways I have explained in the previous video, the one is MAP kinase and other is AKT pathway. Because of these two pathways, it will go and it will cause the phosphorylation of MYC gene, and MYC gene is a global transcription factor for many pro-proliferative genes. It means it will increase the transcription of those genes who are responsible for the proliferation. So it means MYC protein is basically responsible for the cell proliferation. Okay. Now what will happen? After the RAS will attack GTP and it will send signal right immediately within second. What will happen? This enzyme GTPase, it will break this GTP and again convert this GTP into GDP, and this RAS protein will again get inactive. Now it will again get active on the next signal. When the next growth hormone will come and bind to its receptor. It's interesting, very interesting. Now what will happen? This GTPase enzyme is basically inhibiting the cell proliferation by if, for example, assume that there has been mutation in this GTPase gene and loss of function mutation. Loss of function mutation. Now it means this GTPase is not working properly. If this GTPase is not working properly, now it means it will not be able to convert this GTP into GDP back. Now it means GDP will stay with the RAS for longer period of time, and this RAS protein will stay active for longer period of time, and it will keep on sending signal, and it will keep on activating MYC protein. And we know that MYC is responsible for the transcription of lots of pro-proliferative genes. So this MYC gene will keep on expressing those genes, and the proliferation of cell will increase and it may develop cancer. So the loss of function mutation in GTPase can also lead to the cancer. Loss of function mutation in the GTPase can also lead toward the cancer. Clear? The next gene which I am going to talk about is APC gene. Adenomatous polyposis coli. Yeah, I'm right. Here is APC gene. Before talking about the APC, let me talk about the beta-catenin gene. Beta-catenin gene, here you can see it. I have made it red, so it means it is a proto-oncogene. Now what will happen? This beta-catenin gene, the product of this beta-catenin gene obviously will be a beta-catenin protein, and this beta-catenin protein is very helpful. It's also a transcription factor, and this is a transcription factor for this MYC gene. And if this beta-catenin is increasing the expression of MYC gene, it means it will increase cell proliferation. So it will be a proto-oncogene or tumor suppressor gene. Obviously proto-oncogene. Okay. Now what will happen? To counter the beta-catenin activity, there is another gene, APC gene, and the product of this gene is APC protein that will go and bind with beta-catenin, and it will destroy and dissociate it so that overactivation cannot take place. Now assume that loss of function mutation has been taken place in the APC. Now APC will not be able to block this beta-catenin. So now this beta-catenin will keep on activating the MYC, and MYC will keep on increasing the cell proliferation. It means the mutation, loss of function mutation in APC can also lead toward the cancer. And APC is a tumor suppressor gene because it was basically by blocking the beta-catenin, it was basically inhibiting the MYC expression. It was basically inhibiting the cell proliferation. So APC gene is basically a tumor suppressor gene. It is not a proto-oncogene. Okay. Now this is all about tumor suppressor gene and proto-oncogene. I am very much hopeful that you are quite clear about this concept now. Next video will be talking about the nomenclature of cancer and types of cancer. Please don't forget to subscribe my channel and press the bell icon so that you may get notification for those videos as well. And don't forget to like this video and comment and share this video. Thank you so much.